All right, let's get right into it. If you've got a neuroscience quiz coming up, you are in exactly the right place. We're going to break down one of the most foundational concepts in the entire field, the Neuron Doctrine. This is basically the origin story of how we first got a peek inside our own minds.
But before we could understand anything, scientists were facing this huge, kind of ridiculous problem. The brain, well, it has the consistency of jello. Seriously. So how on earth do you study something so soft, so delicate, so complex? How do you map a universe that you can't even slice? That was the mystery they had to solve first. So, to figure out how we finally saw inside the brain, we first have to know what we were even looking for. Let's lay out the basic components of this, well, invisible universe. Okay, so the whole nervous system is really split into two main parts. First up, you've got the central nervous system, or CNS. That's the command center, the brain and the spinal cord. Then, branching out from there, is the peripheral nervous system, or PNS. That's all the other nerves connecting the command center to, the rest of your body. Now, if we zoom way in, this whole system is built from two main types of cells. First, you have the stars of the show, the neurons. They're the messengers, the ones sending all the signals. But then you've got the unsung heroes, the glia. They're the support crew, basically. They nourish and insulate the neurons. And here's a great little fact for your quiz. Glia actually outnumber neurons by about 10 to 1. So let's get back to our jello problem. Even after the microscope was invented, early scientists were totally stuck. They were staring down two massive hurdles. One, brain tissue was just too soft to slice thin enough to see anything. And two, even if they could slice it, all the cells were basically the same uniform creamy color. They were completely invisible to the eye. So how did they do it? How did scientists get past these challenges? Well, they had to invent a whole new toolkit, a really clever process that would finally make those invisible neurons visible. And that toolkit came from a field called histology. It's the study of the microscopic structure of tissues, and it gave scientists the keys to unlock the brain. The histologists were really the detectives on this case. They came up with this brilliant three-step process. First, to solve the jello problem, they figured out how to harden the tissue using formaldehyde. That let them make incredibly thin slices. But the cells were still colorless, right? So the final, absolutely crucial step was to stain them with special dyes. And this staining step was a revolution. Two methods in particular were total game changers. The Nissl stain colored the center, the nucleus of all cells. This was huge because for the first time, scientists could tell the difference between neurons and glia. But the real breakthrough, the real breakthrough was the Golgi stain. For some reason, it only stained a small fraction of neurons, but when it did, it colored the entire cell from tip to tail. And boom, for the first time in human history, we could see the full, beautiful, intricate shape of a single neuron. This image, right here, changed everything. The invisible universe inside our heads suddenly had a face. So what did it show us? It revealed that neurons have this central part called the cell body or soma, and then all these long, thin tubes branching off, which are called neurites. And we quickly learned these neurites have specific jobs. The axon is the output channel, sending signals away, while the dendrites are the input channels for receiving signals. You'd think seeing the neuron would solve the whole mystery, right? Wrong. In fact, it started a massive scientific fight. Now that we could see the individual pieces of the puzzle, the big question was, how did they all fit together? This all led to two rival theories, championed by two scientific heavyweights. In one corner, you had Camillo Golgi, the guy who invented the stain, with his reticular theory. And in the other corner, you had Santiago Ramón Cajal, with his totally different idea, the Neuron Doctrine. Golgi was absolutely convinced that the brain was one giant continuous web. He thought all those neurites were physically fused together, kind of like the veins and arteries in our circulatory system, just one single interconnected superhighway of information. But then you had Cajal. Using Golgi's own stain, he looked at the same slides and saw something completely different. He argued that neurons were individual, separate cells. They weren't fused at all. He believed they communicated by getting incredibly close and touching, not by being physically connected. So remember this, contact, not continuity. That was his whole thing. This became one of the biggest debates in the history of biology. It raged for decades, even after both men shared a Nobel Prize for their work. But eventually, technology would catch up to theory and give us the final proof we needed, I mean, think about how wild this is. Golgi and Cajal shared the Nobel Prize way back in 1906, and there were still fierce rivals. The debate was far from over. It would take another 44 years all the way until 1950 before science could finally, definitively declare a winner. And the winner was Cajal. His Neuron Doctrine, the idea that neurons are individual cells, was proven correct. So how did we finally prove it? With a new, way more powerful tool, the electron microscope. It could magnify images to a level that was just unimaginable before. And with that power, scientists could finally see the tiny microscopic gap between two neurons, proving they communicated by contact, just like Cajal had insisted all along.
And that's the story. The Neuron Doctrine isn't just some dusty old footnote in a textbook. It is the absolute foundation that all of modern neuroscience is built on. It allowed us to understand the brain as a network of individual computers all talking to each other. This discovery opened the door for sure, but it leaves us with a final question. What is the next great mystery of the brain that we need to solve?